Normal shoulder. A coronal T2-weighted MR image demonstrates the normal rotator cuff with the tendon of the supraspinatus, white arrow, appearing as a dark band crossing over the top of the humeral head, HH, and attaching to the greater humeral tuberosity, red arrow. Chronic rotator cuff tear. A coronal T2-weighted MR image in a 64-year-old man shows disruption of the supraspinatus tendon, white arrow, with retraction of the tendon end, located above the glenoid, and atrophy of the muscle belly, red arrow. Fluid surrounds the tendon, bright signal. The humeral head, HH, has subluxed superiorly and articulates with the acromion, AC. Body movements are created by the contraction of skeletal muscles. Skeletal motion is generated by two types of muscle contractions, isotonic and isometric. When an external load is applied to a muscle, indicated by the black bar, the muscle fibers must begin contracting to move the weight. Notice how the muscle torque, yellow bar, is increasing as the muscle contracts. When the muscle torque becomes greater than the torque from the weight, the muscle shortens under nearly constant tension. This is called isotonic contraction. Notice that the muscle bulges, indicating it is shortened. If a larger external load is applied, the muscle torque required increases. The muscle is stretched and the amount of overlap between thick and thin myofilaments in the sarcomere is reduced. As the sarcomeres contract in the decreased areas of overlap, tension develops on the muscle. This tension stretches the tendons and elastic portions of the muscle, but is not great enough to create a muscle torque which will move this heavier load. This is called an isometric contraction. Body movements are created by the contraction of skeletal muscles like the biceps brachii and the triceps brachii. Muscles generate motion by creating torques at the joints they cross. Contraction of the biceps brachii, highlighted in yellow, creates a flexion motion at the elbow joint. Contraction of the triceps brachii, highlighted in purple, creates an extension motion at the elbow joint. Biceps brachii contraction equals elbow flexion. Triceps brachii contraction equals elbow extension. The sites where muscle attaches to bone are known as origins and insertions. The location of the origin and insertion determines the direction of the muscle torque and subsequent joint motion. An example of this is the biceps brachii crossing anterior to the elbow joint and inserting on the radius. This insertion location causes the biceps to create a flexion torque and motion. Similarly, the triceps brachii crosses posterior to the elbow joint, inserting on the olecranon of the ulna. This insertion location results in extension torque and motion. When two muscles act at the same joint and produce opposing torques and motion, they are called antagonists. The biceps brachii and triceps brachii are an example of an antagonistic muscle pair. To activate skeletal muscle, the central nervous system initiates an action potential that travels down the spinal cord to the motor neurons. As the nerve fiber branches, the action potential travels down each branch. Each nerve fiber branches many times and stimulates several skeletal muscle fibers. The union of the axon and muscle fiber is called the neuromuscular junction. Zooming into the microscopic level, each branch of the neuron has a terminal that invaginates the muscle fiber, while remaining outside the muscle fiber plasma membrane. The action potential arrives at the axon terminal. In the terminal, the action potential causes the release of acetylcholine from the synaptic vesicles into space between the axon terminal and the muscle fiber, called the synaptic cleft. In the synaptic cleft, the acetylcholine binds with a receptor site on the fiber membrane, which opens a chemically gated ion channel. Sodium then rushes through the ion channel into the muscle fiber, causing an action potential to form on the fiber membrane. The action potential spreads along the muscle fiber. As more nerve branches activate additional fibers, the action potential spreads over the entire muscle. Upon activation, the muscle contracts. Skeletal muscles, like the biceps brachii, attach to bone via connective tissue called tendons. Muscles are composed of bundles of muscle fibers. Each bundle is separated by connective tissues known as perimysium. Each fasciculus is made up of muscle fibers, which are separated by connective tissue called endomysium. Skeletal muscle fibers or cells are multinucleated and striated in appearance. Muscle cells are composed of subunits called myofibrils. Each myofibril is made up of several myofilaments. The two types of myofilament shown in red are composed primarily of the protein myosin, and a thin myofilament shown in blue composed mainly of the protein actin. The repeating arrangement of thick and thin myofilaments serves as the fundamental subunit of striated muscle contraction. These subunits are called sarcomeres. A sarcomere contraction is represented by the shortening of the distance between the Z lines. The sarcomere shortens because the thin filaments slide past the thick filaments. In 3D, each thick myofilament is surrounded by 6 thin myofilaments arranged in a hexagonal pattern. The 3D arrangement of sliding myofilaments is the microscopic basis of muscle contraction. This is a histological image of cardiac muscle. The plane of this section resulted in a view of the longitudinal axis of the muscle fiber. This histological image of skeletal muscle includes a neuromuscular junction. The visible components include nerve fiber and motor end plate. This histological slide shows skeletal muscle in cross-section. Muscle fibers are separated by connective tissue called endomysium. This histological image contains skeletal muscle. Notice the striations visible in the muscle fiber. This histological image contains smooth muscle, which can be found in the walls of hollow organs, including blood vessels and the digestive tract.